Alright, so what we're going to do here is we're going to break up chapter 14 notes into a couple of different chunks. The first chunk is going to be history, discovery of DNA, that type of thing. Alright, so here we go. So obviously, I know some of you guys had questions about this. Justin, I think you did, Griffith's transformation experiment. So what we have going on here, pretty simple. We've got a live virulent strain of pneumonia. It's going to kill the mouse when you inject it. Then there's a live non-virulent strain. Now obviously if it's alive but it's non-virulent, you would expect that the mouse should live. Then they did a heat-killed version of the virulent strain. So notice the polysaccharide coat remains intact, but what's going to happen is the DNA or the RNA inside the virus or the pneumonia is going to be fundamentally altered. Therefore, when you inject it, the mouse should live. Now, here's the next part. Okay, we mix these two together, the live non-virulent and this heat-killed virulent. And what happened is something got basically moved from one of these to the other. Okay, so there was this movement of material, there's this movement of information. And this is when we kind of started going, okay, so what is being moved from point A to point B? There was some kind of chemical that had to have moved from one to the other. And this is where we get the idea of transformation of DNA, and DNA being the thing that gets moved from point A to point B. But they didn't know that initially. Okay, so they didn't know that. All we know is that something had to go from this guy to this guy, and that's going to be our transformation material, which turns out is DNA. Now, they had to do some more information and more research to figure this out. So along comes Hershey and Chase, and they did an experiment. And the big thing at the time was, all right, well, what's the thing that's getting moved around? What's the information that's moving from the live strain to the non-virulent strain? And there were two ideas. It's either proteins or it's either DNA. Most people thought proteins at the time because they're much more complex. DNA only has those 4 base pairs, so they were like, it's not really that complex, that's probably not what's going to be getting moved. So many people thought proteins. So what they did was they used a bacteriophage. Now bacteriophages, this is a virus that eats bacteria. It basically has 2 parts. It's got an outer coat and it's got the inner DNA. Now the outer coat is made of proteins. The inner material, DNA, obviously we know what that's made of, nucleotides. So what they did was they grew these bacteriophages in 2 different types of cultures. One was in a heavy sulfur, and we know that sulfur is going to be incorporated into the disulfide bridge cross links in our proteins. So the sulfur is going to get incorporated into the outer portion of the bacteriophage. They also grew some on heavy phosphorus. Now phosphorus, we know that phosphorus is going to be used in our sugar phosphate backbone in the DNA. So here we have DNA that is now irradiated, and here the outside shell is irradiated. So what we're trying to say is, well, how does this virus hijack a bacteria? What is it transferring? Is it transferring its protein outer coat, or is it transferring the DNA? And so by labeling these things radioactively, we could figure out which one was doing what. And what we found out was that the thing that got transferred and kind of hijacks the bacteria is the DNA. So when they actually then took these bacteria and they crack them open and they separate the phage coat from the bacteria in the blender, and then they centrifuge it, what they found was that in the supernatant, which is the liquid that gets separated, is that's where they found the sulfur, which means that pretty much all of the protein stuff never got transferred into the bacteria. Okay? Down here we have the 32 phosphorus pellet, and that is what got transferred into the bacteria when they crack it open. So we can actually see, using the radioactivity, what moved into a cell and what didn't move into a cell, and that led us to understand that DNA is going to be this transforming agent. It's the thing that gets passed from generation to generation. Now, you guys know what this is. You can see, right, there's going to be my nucleotide. I'm pretty sure you know how to count 1, 2, 3, 4, 5 carbons. So sugar phosphate backbone, and here's going to be our nitrogenous bases. Again, they're going to link together with that phosphodiester bond up here. And we should, I'm hoping, we know the differences between the nitrogenous bases in terms of purines and pyrimidines. Remember, purines are 2 rings, pyrimidines are 1 ring. And then the difference between thymine and uracil, one's found in DNA and one is found in RNA. Simple enough. All right. Now, how did we get to this part? There's some people we got to talk about. One guy, Erwin Chargaff, he was basically a biochemist. He's the guy that came up with the base pairing rules, so A to T and C to G. Maurice Wilkins and Rosalind Franklin, they worked together in X-ray diffraction labs. Rosalind Franklin was the one that basically discovered that if you take a picture with x-rays, you see this X shape, which must mean that the DNA is twisted into a helical type of shape. Wilkins, he kind of took some of her pictures and gave them to Watson and Crick. They were kind of stuck at a certain point. They couldn't get the molecular geometry of DNA right, and what they did was they built a model and wrote a 2-page paper, and they ended up winning a Nobel Prize in 1953. Wilkins also got a part of that. Rosalind Franklin was dead because of working with x-rays all the time, and Erwin Chargaff was left out in the cold, and he was very bitter about that. All right, so we know that what's going to happen here is we have our nitrogenous bases. If you have G and C, you have 3 hydrogen bonds. A to T is 2 hydrogen bonds. This is clearly going to be more difficult to break because you have 3 bonds. Here's the picture of what Rosalind Franklin did as far as the X-ray diffraction pictures that she was taking. You can see that X, which that's going to tell you that obviously you know, the DNA is helical, it has to overlap. And over here we've got Watson and Crick, and this is the famous model that they built, and they wrote the 2-page paper, and they won the Nobel Prize for that. All right. Our current model, there's all sorts of different types: ribbon, ball and stick, space filling model. I'm sure you've all seen this before. This is nothing new. Be aware that there are minor and major grooves here in DNA that'll play a role into some things that we can do to kind of turn DNA on and off. So that'll be important come later on. Obviously, anti-parallelism. What you're looking for to determine which way is facing up. If you say phosphate, that's the 5' side. 5' is the phosphate pointing up. 3', it's on the 3' carbon. So 1, 2, there's the 3rd carbon. You're looking for that hydroxyl group. So that side's facing up. This side is pointing down. There's my 5' phosphate and my 3' hydroxyl. That directionality is going to be important, especially when we get into the replication stuff. So make sure that you understand that. The directionality will be huge for replication. All right. As far as the replication models, you guys already wrote about this. That was one of your Pitt Bio assignments, was to talk about like which model of replication is the right one. There's the conservative model, the semi-conservative, and the dispersive. And I think we all know that the semi-conservative model is the one that we should be looking at, and that was based on the Michelson-Stahl experiment, which we understand how this works. Again, heavy nitrogen, light nitrogen. As it starts to incorporate the light nitrogen isotopes, the mass of the DNA is going to get less. So as you spin it, you're going to have less mass, so you're going to have less travel, and you're going to end up getting these different samples here. So we know that it's a semi-conservative model of replication based on this very simple experiment. Now, as far as the one last thing I think I want to touch on in this little chunk before we move on to anything else is DNA polymerase and RNA polymerase. What these guys do. Now, DNA polymerase is going to be the enzyme that's going to be responsible for replication. Its job is when it sees an A, match it with a T, a C, a G, and so on and so forth. Now, here's the thing about DNA polymerase and RNA polymerase. These enzymes only like to work in one direction. These enzymes like to work in a 5' to 3' direction. So what they're doing when they build, right, here's 3', there's 5', they build 5' to 3'. So the DNA polymerase is literally going to be adding things on, and it's going to be looking right there every time for that 3' hydroxyl. If it can find a 3' hydroxyl, that's how it's going to know, oh, I'm going to add another base in there. Now, as it does this, right, we can see what's going to happen. We got to clip off our 2 phosphates. So again, it's going to be a dehydration synthesis reaction, right? We're going to have to basically clip some water out there and then put these guys together in there. So it's going to be that kind of reaction. DNA polymerase has 2 major jobs. 1, it writes new DNA, and 2, it goes back and proofreads its work and makes sure that it's doing everything correctly. Because if your DNA gets messed up, that's going to be problematic. And that's where I wanted to stop. All right.